Hello, everyone, and welcome to today's webinar, FlowCodes, a flow cytometry-based platform for massive parallel in vivo screening. I'm your moderator today, Aaron Tyznik, and I have the pleasure of introducing our webinar speaker, Adrian Liston. Adrian is Professor of Pathology at the Department of Pathology, University of Cambridge, along with numerous affiliations, too many to list. Adrian, along with James Dooley, actually run a laboratory focused on immune regulation and responses with particular interest in the brain, but also has been a pioneer in a lot of different aspects of flow cytometry and utility of those tools, which we'll hear about today. I personally had the honor to work with Adrian when we both had a couple positions at the University of Washington, and have continued to follow his work over the years. So we will have time at the end for some Q&A. So please place your questions in the correct chat box at any time during the webinar. Keep in mind there is a chat box, but then there's also a question and answer. Place your questions in that particular Q&A box. I'd also like to note that the webinar is being recorded and will be available within a couple of days on CytoU. Everyone, please welcome Dr. Adrian Liston.
Thank you very much, Aaron. It's a pleasure to give this talk. And let me just share my screen. Okay, so what I want to talk about today is a talk that is half about the biology of tissue-resident regulatory T cells, or Tregs, and half about the flow cytometry tools that we use to do massive multiplexing of in vivo experiments. So if you're interested in the immunology side, there's something for you. If you're interested in the flow cytometry development side, there's something for you. And if you like neither immunology nor flow cytometry, then I question your choice of seminars to come to. Okay, so just to get started, we're talking about regulatory T cells, also known as Tregs. And the type of regulatory T cells that I'm talking about are the FOXP3 positive regulatory T cells that typically are generated in the thymus and are very immunosuppressive. So this population of FOXP3 positive Tregs, they're known to be absolutely critical to shut down immune responses. They prevent autoimmunity, but they also regulate the strength of anti-cancer responses. They regulate the response against infections. And so the right number of regulatory T cells in the system is absolutely critical for how well you've got this balance between immunity and tolerance. Now, almost all research on regulatory T cells is based on this population that is present in circulation, in the peripheral blood in humans, or in spleen and lymph nodes in mice. However, there's a growing appreciation that there are also tissue-resident regulatory T cells that are present in basically every organ. It's thought that these regulatory T cells are required for the local immune control, so the strength of their immune responses inside the tissues. Well, actually, that is a very difficult question to show experimentally, because sometimes it's difficult to separate out the role of the regulatory T cells inside the tissue compared to the role of the regulatory T cells inside the draining lymph node. We don't always know where the regulation of the tissue response is happening. One thing is becoming clear, though, is that there is an importance of these tissue-resident regulatory T cells beyond simply controlling immune responses. These cells are able to alter normal physiological processes. For example, the cells in the adipose can improve insulin sensitivity. The cells in the muscle can aid muscle repair. In the brain, they can promote oligodendrocyte differentiation and promote remyelination. So as well as simply quenching down immune responses and shutting down the immune response, they're also able to activate and accentuate this repair and regeneration process inside the tissues. Now, until very recently, the model for tissue-resident Tregs is a model that I'll call the tissue seeding and specialization model. Under this model, the regulatory T cells are going into the tissues at an early age. At that point, they're specializing so that a muscle Treg and a a lung Treg will end up looking quite different, and they have this indefinite residency inside the tissues. Now, this model was largely built up as an analog to the model for tissue-resident macrophages, because indeed macrophages enter the tissues very early. They really specialize unique flavor per tissue, and they reside there indefinitely. But what we asked is this model that was put onto tissue Tregs from the macrophage world, is it actually something that we can test empirically by the data? And so we started off by doing high parameter flow cytometry just to look at the phenotype of the regulatory T cells. Until this point, people were looking at tissue resident Tregs compared to, say, the spleen or the lymph node. And you could say that the Tregs in the muscle were different from the Tregs in the spleen, and the Tregs in the liver are different from the Tregs in the spleen. But we weren't doing side-by-side comparisons of multiple tissues. So here what we did is we took 48 different tissues, we perfused the mice, and we used anti-CD45 intravenously injected so that we could exclude any vascular contamination. And then we ran a 46-color spectral flow cytometry panel. Now, what we've got on the right is a meta-PCA, which is looking at essentially the phenotype of the Tregs in each of those tissues and clustering how similar those phenotypes are. And what we'll see is that in the green, all the lymphoid tissues are clustering. So Tregs in different lymph nodes and in different spleen and thymus and so forth, they all look very similar. In yellow, you've got largely the gut-associated Tregs, and they're all quite similar. Although I should note that really it's more about the small intestine Tregs being very similar. The samples at the border are the large intestine samples. And we've got a few outliers like the bone marrow and the blood and peritoneum. They look a little bit different. But by and large, if we look in purple, those are the non-gut, non-lymphoid tissues. They all have a very similar phenotype. And in fact, they have more or less the same subsets of Tregs are going to be present right across the body. And what's different is the ratio of a couple of key different populations between the different tissue sources. So in the blood, you'll have a lot more naive Tregs. In lymphoid tissues, you'll have a lot more generically activated Tregs. Then you have these additional populations that show up in the tissue that are tissue resident in their phenotype. And in the gut, you'll have some of those, but you'll also have a kind of a unique population that's got more of a gut phenotype. And I'll get into those phenotypes later on. But I do want to make the point that it's not that the whole population is different phenotype in different tissues, but rather the ratio of these subpopulations differs in the different tissues. Now, flow cytometry is our mainstay, and I think it's an incredible tool. There is, however, an intrinsic limitation in that you get information based on what you look for information about, what antibodies you choose. So we also did we also did bulk RNA sequencing and single cell sequencing, but this is the bulk data, and we can purify Tregs from different populations and look at genome-wide transcriptional profile. I'm not going to go through this data in detail because it's actually quite boring. The Tregs from different tissues are remarkably similar. You look at the Tregs in the kidney and the Tregs in the pancreas, and their transcriptome is 98% identical. There's some genes are a little bit higher and a little bit lower, but there are no unique genes that separate them out. Now, if you look at the gut, they start becoming a bit more different, maybe 10% differences across the transcriptome. But if you're interested in this data, you can use that QR code or use the link, and you can go and play with our interactive viewer where you can pick the organ or pick the gene and look at the transcriptional signature. Now, we next want to ask whether or not the Tregs are indefinitely staying inside the tissues. And so for this, we did a parabosis experiment, CD45.1, CD45.2. You can just consider them red and blue cells for this purpose. You surgically join the mice together, and the blood vessels join up very quickly. And so now any cells in the blood are rapidly circulating between both of those mice. So you now have parabolic pairs, and you can take down those pairs at different time points, and you can look at multiple different tissues. And essentially, you can ask, what is the rate at which cells in the tissue are displaced by cells coming in from the blood? And just to really cut a lot of analysis very short. Essentially, it's the activated Tregs that can enter the tissues, but about half of them are leaving and dying very rapidly, within days to a week. And these are probably the Tregs that come in and then they don't see any of their antigens. However, about half of the Tregs are coming in and either they're already CD69 positive or they're turning on CD69. And this is the marker for seeing their own antigen inside the tissue. And at this point, if they turn on CD69, then they tend to stay a little bit longer in the tissue, about a median of 3 weeks in the tissue. And at that point, they either leave or die. So we can look at this population of CD69 positive Tregs. So these are not just all the Tregs in the tissues. These are the Tregs in the tissues that have got the longest dwell time and have got the most tissue specialized phenotype. And if we can calculate the median dwell time for these CD69 positive Tregs in cross different tissues. And what we'll see is that essentially it's about 3 weeks across multiple different tissues, a little bit longer, a little bit shorter in different tissues. You know, the lung is on the short side, while the brain is a bit on the long side. The outliers are the white adipose tissue and the different layers of the gut. But even there, we're talking about 10 weeks. So the tissue residency is really on the order of weeks to months. It's not on the order of the lifetime of the organism, the way it is for tissue macrophages or for NK cells in some tissues, for example. So if we go back to this model, we've got this tissue seeding and specialization model that we've adopted from macrophages. Remember, the key features are a distinct wave of seeding, unique specialization, and indefinite residency. And it doesn't look like any of these hold up. We don't see these features in the tissue-resident regulatory T cell population. So the model that we generated is what we call the pan-tissue model. Now, under this model, there is a circular migration of Tregs going through the system, and some of those Tregs in circulation are able to enter tissues. When they enter tissues, they turn on a tissue residency program, but it's a pretty generic program that is fairly similar from tissue to tissue. And the residency inside the tissue is pretty transient. It's on the order of weeks to months. Now, we will have one small exception in that the gut does look a bit different. In fact, there's a subset of Tregs that go to the gut that look different, while most of the Tregs in the gut do look like Tregs in other tissues. So that's the model. How do we test it? And this is where we're getting into more technology-driven work using flow cytometry. Now, the first test of the pan-tissue Treg model is if this model is correct and regulatory T cells are able to go, the same cells are able to go into multiple different tissues, then we should see the same TCR coming up in multiple different tissues if we look at the Treg population. And the reason for this is that the TCR, the T cell receptor, is a genomic barcode. It's recombined during thymic development at a very early stage, and it's combined at the genomic level. So when that Treg is formed and it proliferates a couple of times, all of those daughter cells that recognize the same antigens, they all have this same genomic sequence on the TCR. So if we pick up that sequence in multiple different tissues, we can tell that those clones, the cells from the same clone, have gone into different tissues. Now we can test this question by extracting Tregs from different tissues and then performing single cell sequencing using 10x VDJ-seq, which means that we can sequence the alpha and beta chain of the TCR at a single cell level. And this gives us the type of chord diagram that we see here, where the outer ring are gut Treg TCRs from different tissues. And so we can see some cells from the gut and the liver and the pancreas and so forth. Then the middle ring is the different mice. And so here there are 4 different mice in this experiment where we took the different organs from the different mice. And then the links between it inside the center, that is saying that the same TCR is being found across different samples. Now, what you can see here, we've highlighted all of the gut Treg TCRs. So this is the sequences found in Tregs inside the gut. And you can see some sharing from mouse to mouse. And you can see a little bit of sharing from the gut to other tissues. So sometimes you'll find the same clone will be found in the gut as is found in the kidney and in the liver. But generally, not a whole lot of sharing. Now, this is quite different if we look at the liver Tregs. So here we've got the liver clones, and we can find a lot of liver clones present in the kidney and the pancreas and the blood, but we don't find many of those clones inside the gut. And so through a process of normalization, essentially what we can say is that the sharing between kidney, liver, and pancreas is all at about 60% of the sharing that we'd expect just inside a single organ. Now, this means if you take out one kidney and you find a Treg inside it, that Treg is almost as likely to be in the liver as it is to be in the other kidney. Now, if you look between the gut and these organs, the sharing rate is about 10%. It's pretty substantial sharing still, but it does mean that there is a distinct population difference between the gut Tregs and the non-gut Tregs. Now, the second test for this model is that we should be able to confer a multi-tissue homing pattern on regulatory T cells by giving them one of these tissue Treg TCRs. So just to explain this a little bit more, the idea is that if we take a tissue Treg TCR, so we're taking a regulatory T cell that's living inside a tissue, we sequence and clone that TCR, and then we force a regular T cell to express that TCR, we can then look at the behavior of that T cell that's now got a transgenic TCR. And we can measure its fate. We can say, did that transgenic TCR force the T cell to become a Treg? And if it became a Treg, did those Tregs go into multiple different tissues, or did they go into a single tissue? Now, this is the type of experiment that has been done. It's called retrogenic TCRs. It's a type of experiment that's been done before, but typically you only analyze one TCR because it's time-consuming and expensive to create a TCR transgenic mouse. The problem with that approach is that you just don't know if your TCR is representative if you've only done one. So we tried to multiplex this assay up using the FlowCodes system. Now, just to explain what the FlowCodes system is, it's a derivative of the ProCodes system built by the Brian Brown lab at Mount Sinai. Now, what they've got in the ProCodes system is a way of barcoding retroviruses, or in their case, lentiviruses, so that you can couple the cargo of a lentivirus with a barcode that you can read out at the flow cytometry level. Now, this is very similar to having one retrovirus expressing gene 1 and RFP, and one retrovirus expressing gene 2 and GFP, and you can put those into the same mouse, and you can read out what those two different genes do by looking at RFP versus GFP. Now, the problem is that you can only build that up so high before you've reached the end of your fluorophores and you've maxed out your panel before really adding any of your biology. Now, the great thing about the ProCodes is that they're only using 14 different antigen tags. And these are things like HA tag and V5 tag that everyone can get antibodies to. But they create linear epitopes with 3 antigen combinations. Now, this means with 14 different antigen tags, you end up having 364 unique combinations of 3. And so now each of your viruses can have a tag and, in this paper, a guide RNA, and you can read them all out using cytometry. Now, the original paper was validated in vitro using Cytov. And unfortunately for us, when you move this system in vivo and with flow cytometry, you end up with quite a lot of problems because the original tags are fused to the surface of the cell, so the cells get rejected. And even if you solve that issue, the signal is too weak to pick up by flow cytometry. So it took us a couple of years, but we took the basis of this ProCode system and we adapted it for flow cytometry, and in particular flow cytometry in vivo. Now, the way we did this is we used those same antigen tag combos. So it's the same triplets of 14 different tags, but we fused them to histone 2b. Now, there's a lot of advantages to fusing to histone 2b. One of those advantages is that because you've got an intracellular tag, you're preventing rejection in vivo. So we've tested, and these cells survive in vivo a long time with no problem. The tag also, because it's fused to histone, which is a very long-lived protein, it builds up to much better levels for detection by flow cytometry. So you can get good signals for this tag by flow cytometry without really struggling to detect the tag. And then finally, we can use intracellular staining for markers like FOXP3 to delineate Tregs without getting any loss of the tags, because the permeabilization does not release the histone tags. And we've now validated this approach in vivo with flow cytometry. We also had to build our own R-Shiny to analyze this, essentially to decode which triplet each cell has, and that's available now online, FlowCode decoder built by Adrian. So we can now go back and take our different T cell receptors, and we can take a set of different T cell receptors. They're now going to be our cargo, and we can put them into different retroviruses. And each retrovirus has a unique TCR cargo, but also a unique flow code. And we can then read it out with these different antibodies, which triplet is present, and therefore which cargo that T cell is carrying, that cell is carrying. Now, we're now doing sets of 100, 200 different cargoes, but in this first experiment that I'm going to show you, this is a set of 20 different codes. So what we do is we take our FlowCode plasmids, we create retroviruses, and we actually make our retroviruses in 96-well plates, and then add those retroviruses into hematopoietic stem cells on 96-well plates. Now, the reason we do this is because if you just do bulk production and bulk transduction of your hematopoietic stem cells, you get quite a lot of variation in the frequency of those codes. By doing everything individually on 96-well plates and then pooling the population afterwards, we get very even representation. So we've now, each of those retroviruses, each of those hematopoietic stem cells, they all have a unique retrovirus, which means they all have a unique T cell receptor. And then we bulk it up the population of some wild type cells, and we transfer into an irradiated mouse. Now, this means we now have a mouse where about 80% of the T cells are wild type and polyclonal, and the other 20% are constituted by more or less 20 different TCR transgenics at 1% frequency each. So they're all low frequency, but high enough that we can measure their frequency as individual clones, and we can trace them all by flow cytometry. Now, this is the, you get these huge data sets that come out of simple flow cytometry experiments once you do this. And this is, it's a bit of a complex data set to look at. So if we just concentrate on that top set of circles, what these circles are telling us is whether or not the cells stayed as conventional T cells. So in that case, they're blue on this diagram, or if they became Tregs, in which case they're green. And if they became Tregs at very high rates, they will be yellow or orange or red. So the physiological rate of Treg conversion is about 25%, and they'll be green. Now, if we go across the slide, we are looking at the 20 different clones, and 10 of those clones are TCRs that we originally found in multiple different tissues, and 10 of those clones are TCRs that we originally found in only one tissue. And if you just look at that sea of green and yellow and orange and red, what you'll see is that the majority of these clones are becoming Tregs at normal levels, green, or very high levels, being yellow, orange, red. They're also becoming Tregs And then the size of the bubble is how many cells that we're actually counting in this set. Now, if we look at the bottom row here, now this is saying of the Tregs that get into tissues, what fraction of them are found in each of 4 different tissues: the kidney, the lung, the liver, and the pancreas. And what we see is that generally these Tregs are actually going in to multiple different tissues and are found in multiple different tissues. Now, there are 5 exceptions. These are 5 TCR sequences that when we force a Treg to express them, they go into the tissues, but more than half of the Tregs end up in one particular tissue. And interestingly enough, these exceptions come from the sequences that we originally found in one tissue only, and it correlates to finding it in one tissue only. So if we look at Treg, tissue Treg TCR12, that was originally found in the kidney, and what we find is that more than 75% of the Tregs go to the kidney. So it's kidney biased. We'll also see that a couple of the gut Treg TCR sequences are very gut biased, while one seems to be quite happy to go into multiple different tissues, and a couple of the pancreas only sequences end up being pancreas biased. So what this tells us overall, of those 20 different TCR sequences we analyzed, all 20 are enriched for becoming a regulatory T cell, and all 20 become multi-tissue in their migration. However, of those 20, 5 of them have bias towards the tissue of original detection. So if we had to make a rough estimation at this point, and I really do want to say rough, we would say that perhaps 75% of the tissue Tregs are multi-tissue and 25% are tissue biased, but not tissue restricted. Now, the great thing about the FlowCode system is that we can now use this approach and multiplex even higher and test hundreds more. And I really think that when we are analyzing regulatory T cells, or for analyzing TCR repertoires, we need to be looking at analyzing hundreds of sequences before we start trying to draw conclusions. Now, the third test of this pan-tissue model is a relatively simple one on rehoming. And what this test is that if we take regulatory T cells out of the tissue and then we inject them back into a mouse, do they go back to the same tissue, which suggests that they're tissue specific, or do they end up going back to multiple different tissues? And so here Oliver ran a really nice experiment. We would now probably do this with FlowCodes, but at the time we did this with congenic mice. And what we want to do is take Tregs out of multiple different tissues and then pull them together. So we had a single pool with Tregs from multiple different tissues and track their fate afterwards. Now, the way we did this was to have multiple different congenic mice. So CD45.1, CD45.2, CD45.12, GFP CD45.1, GFP CD45.2, and so forth. So by combining congenic markers and fluorescent markers, you can easily get 8, 10 different pools of mice. You then take organs from each of those mice and you mix match them so that the liver Tregs will be from CD45.1 and the lung Tregs will be from CD45.2 and so forth. So we can track, after transferring them into a new mouse, we can track where the Tregs originally came from based on their congenic marker and where they ended up being based on which organ we're analyzing by flow cytometry. And if we do this, we can calculate a migration index to say, are the Tregs from those tissues likely to go back and rehome to the same organ? Are they avoiding the organ they came from, or are they more or less agnostic? And what we'll find is that Tregs from the gut tend to be biased to go back to the gut, so they rehome into the gut twice as likely as chance. Maybe a little bit of bias for bone marrow Tregs going back to the bone marrow, but kidney, lung, liver Tregs, these are really just as likely to go back to a different organ as they are to go to the same organ that we extracted them from originally. So the conclusion for this pan-tissue model of Tregs is that the Tregs in the tissue are really in a general circulatory migration pattern. They can enter tissues. At that point, they will turn on a generic tissue residency program. So tissue Tregs look different from the spleen, but a Treg in the gut, sorry, a Treg in the liver, Treg in the lung, Treg in the pancreas, they look more or less the same. And this residency is transient, about 3 weeks to a couple of months, depending on the organ they go to, with an exception that the gut Tregs do look a little bit different. Now, just before I end up, one thing I'd love to be able to tell you about, and I can't tell you the answer because we don't have it yet, but I can tell you how we're approaching it, is what are the molecules used by Tregs to get into the tissues, and are they using the same molecules in different tissues, or are they using different molecules for different tissues? Now, actually, there's a relatively discrete set of migratory molecules that are available for a cell to enter the tissues. You know, you've got integrins, adhesions, cohesions, selectins, chemokine receptors, chemotactic receptors, but overall, it's about 150 genes. Now, this puts it in the realm of a FlowCode library. So we can take guide RNAs against each of these 150 genes and clone them into 150 different unique FlowCode retroviruses, put those retroviruses into Cas9 cells, and then pull them in a mouse. And essentially, at that point, you have 150 different knockout cell lines or primary cells in a mouse, and you can look at their behavior and read it all out by flow cytometry, by unmixing that barcode based on the expression of the FlowCode barcode. So this is the type of experimental result you get. Here we've got 10 mice and 18 tissues, and that's a relatively manageable 180 FCS file experiment. But what that experiment has is data for each of 155 guide RNAs, and what is the impact of knocking out that gene on entry into each of 18 different tissues, because we can look at Treg frequency in the tissue versus Treg frequency in the blood or the spleen as a reference. We can also look at different subsets. So we're interested in Tregs, but from that exact same experiment, we've got data from conventional CD4 cells or from CD8 cells, and you can use this library in any cell type you're interested in migration. We can also look at whether or not the guide RNA impacts the phenotype. So we can do a t-SNE plot of the knockout versus wild type cells, or we can look at the ratio of knockout and wild type in activated memory and naive subsets. So maybe the central memory CD8 cells are using a different molecule than the effector memory CD8 cells, and we can work it all out without doing a new experiment. We just run our gating differently. And we get these type of heat maps. It's a heat map of all the different genes and all the different tissues for Treg entry into the tissue. And it's a functional heat map, not an expression heat map. And I think that's a real power of this approach. Now, like I said, I can't really show you any data because we're just starting to analyze it. But this is the type of effect that you can get. This is Tregs in the lymph node versus Tregs in the spleen. And you can look at some guide RNAs are going to pop up in the lymph node and samples as being enriched and some in the spleen. And that means that these molecules are involved in entry into the lymph node, and these molecules are involved in entry into the spleen. And you can really unravel that type of biology. This is another example of looking at entry into the gut versus entry into the spleen. And this is kind of a nice little example, actually, because ITGA4 pops up as being important in entering into the gut. And then we can look at ITGA4 frequency of the barcode, and you see it's quite high in the spleen, it's high in the lymph node, in the gut and the Peyer's patches, it's quite low. And actually the mesenteric lymph node, it's intermediate. And we're now doing some tests, but it looks like that expression is required to get into the mesenteric lymph node, not directly, but the cells that go to the gut and then leave the gut via the mesenteric. So there's a partial effect that's being measured there. So just to finish up the talk, I think FlowCodes are a really useful approach for anyone who wants to do massively parallel flow cytometry experiments, and in particular, massively parallel in vivo flow cytometry experiments. Because essentially what we're doing is single cell barcoding that we're reading out by flow cytometry. The great thing about this is, it's cheap and it's fast to get data on millions of cells. Compare that to Perturb-seq, where 100,000 cells will bankrupt the lab. You can literally run 100,000 million cells without a problem by flow cytometry. It also has very high recovery, unlike CyTOF or unlike sequencing-based experiments. So it's great for rare populations. We can barcode in CRISPR guides for targeted libraries. Now, we can't do a huge library, but hundreds to thousands of genes you can do CRISPR libraries for. We can barcode genes, so we can track hundreds of T cell receptors or hundreds of B cell receptors simultaneously. It's compatible with image stream, so that we can look at subcellular localization. You can screen for genes that are changing whether or not your marker is in the nucleus or in the cytoplasm. And it's compatible with intracellular readouts such as FOXP3 expression. Now, of course, it's not a panacea. It's a maximum of 364 barcodes per library at the current depth. That's pretty straightforward, I think, to get into the low thousands, but it is going to reach a limit. And every new library requires individual cloning of retroviruses. So it took us, now it takes us a couple of months to make a new library, but it does take that investment because you're cloning each one individually. So if anyone wants to study migration, we'd be very happy to share our library because we went to a lot of work to make it, and we're very happy to share that. And then finally, it requires access to either spectral flow cytometry or to CyTOF. You really struggle if you're doing this on regular flow cytometry because you've already eaten up, say, 10 to 14 different channels just with your flow code tags before you start doing the biology. Now, before I finish, just to mention, our flow cytometry guru is Oliver Burton. He's fantastic. He knows everything about flow cytometry. I really highly recommend that if you're interested in doing ultra high parameter flow cytometry, and he's now getting up to 70 or so markers, read his blog, Colibri Cytometry. It goes in depth into dealing with different issues. And I've also put some QR codes there for different flow cytometry resources the laboratory has made, some wet lab protocols, some analysis protocols. And with that, I'd like to finish up. Really want to thank Oliver, Oriane, and Magda, key drivers of this project. James and I run the lab together. Key collaborator Brian Brown for sharing the ProCodes in early stage with us. QR link to the data sets, a QR link to some career advice articles. The lab does a lot of career development work. And thank you to the people who funded us.
All right, great. Thank you so much, Adrian. Absolutely astounding being able to take this technology and expand the capacity of what you can do. We have a few questions here that people are kind of curious about. One which you actually mentioned at the very end. So, are these retroviruses libraries available for others to do, and that they'd have to clone them in? But it sounds like that's something that you're making available for other researchers if they want to pursue these types of studies.
Absolutely. I don't believe in reinventing the wheel. Absolutely want to share these libraries far and wide. We are putting the empty libraries on AdGene right now. So as soon as they become available on AdGene, you can get them from there. At the moment, you got to get them from us, but hopefully in a couple of weeks, they'll be on AdGene. And then if you want the libraries that we've already cloned things into, like the migration factor library, just contact us and we'll send you over the plasmids and you can grow up your own retroviruses.
Yeah, that's astounding. A true spirit of scientific community. I really, really appreciate that. It makes me even think about when some prior studies where people had looked at migration with the best tools that people had at that time, which would be maybe parabiotic studies looking at memory cell migration and so forth. And so just to get a sense of this and the scale of it, how many mice have you actually been able to kind of perform? Like I know the migration studies, but the Treg studies that you've actually been able to run through this process with all these various T cell receptors.
So we tend to do experiments of about 10 mice for a library. That starts to become then quite robust, and we find that adding more mice after that doesn't really solidify the results. Now, the key thing is really matching up the size of the library to the number of cells that you're looking at. So if you're looking at relative migration between the spleen and the lymph node, you got millions and millions of cells, you can have an enormous library, and you'll get very stable results. But if you're looking at migration of Tregs, which is a small number, from the spleen to the brain, you've got maybe 100 Tregs that you're going to recover in the brain. If you try to do a library of 500 genes in the brain at that point, it's going to be incredibly stochastic and noisy. And I'd recommend you just don't do it. Just break your library down into 10 or 20 at a time. So it's really just about matching the size of the output files to make sure that they are, on the same order of magnitude as the input library size. But typically speaking, if you match that up about right, 6 to 10 mice gives you pretty stable results.
Yeah, great. Appreciate that. Another question here regarding, there's always a lot of, you alluded to it right there, where you may only get 100 Tregs out of a brain versus, as we know, spleen, lymph nodes, very robust, even the gut. Does your lab offer, and perhaps maybe this is a part of the work that Oliver has done, protocols that kind of help people standardize their processing to effectively recover cells? That's the biggest challenge is, the inconsistency of people's secret sauce to make liberate the cells, and then a different set of researchers may do it a little differently. And if things aren't available, any guidance on how you handle and approach this?
Yeah, absolutely. This is key. It's hard to talk about a universal protocol, I have to say. We work a lot on the brain, and the best protocol to get out regulatory T cells is not the best protocol to get out microglia, and that's not the best protocol to get out neurons or astrocytes. So it does matter a little bit about the type of cell type you're getting out, and then the type of tissue. So protocols in the gut and the brain and the bone marrow are all slightly different protocols. All of our protocols are open access and shared. Although, there's a big difference between making a protocol accessible and people actually accessing it. Even inside our lab, sometimes I have to convince people to just use the protocol that's already available rather than just winging it yourself. That's always the difficulty, is the adoption of a good protocol.
Yeah. Appreciate that. We have a couple also I would describe as more immunology, biology-based questions. And one was when we think about the Treg tissue and the resident pattern, I know that many studies, and there's only so much you can do. Have you had an opportunity to look at like post-tumor challenge or infection, and do these, or are there other models like autoimmunity and so forth, where you can maybe look at these potential changes in the context of disease when we think about responses and so forth?
Yeah, best question. I mean, I am sure that what happens in homeostasis is going to change with disease, and probably change with different types of disease. This is an area where we're starting to work on now. We don't have any data, and then we're going to really focus in on the lung, the gut, and the brain being the type of axis that we're interested in. But really, this needs to be what we would ideally have is an understanding of the behavior in health, and the understanding of the behavior in 5 different inflammatory models across each individual organ. An incredible amount of work that needs to be done. But the more that we can standardize those type of experiments and we can do those cross comparisons, the better our understanding will be at the end. So we're moving in that direction, but it's a long way and there's only so much that we can do.
Yeah, for sure.
So again, this is why we're happy to share our tools and to collaborate with anyone who is interested in a place that we'd love to know the answer to, but we're not going to go ourselves.
Well, I was just thinking about it, like as you mentioned, your kindness in the scientific community to share these retroviruses that are already preloaded with some various Treg TCRs that people can start applying this to their particular model system. And that leads to another question for those who aren't overly experienced in Tregs, Treg biology, Treg function. Maybe could you expand a little bit more on that aspect of the TCR for Tregs and why that could be driving that tissue homing or, well, traveling to and some staying but others leaving?
Absolutely. So the TCR is very similar to BCR or immunoglobulin, which is antibody. So each TCR can recognize one antigen, the same way each antibody can recognize one antigen. TCRs are a little bit different in that they don't recognize free-floating antigen. They recognize antigen that's being presented to them on another molecule. For Tregs, that's MHC class 2 or HLA class 2. And what this means is that each Treg, when it develops, it's really good at seeing one molecule. Now, let's imagine that one molecule the Treg can see is actin. Yeah? Actin is presented everywhere across the body. There are actin peptides presented. That Treg will go into any tissue. It'll see actin. It'll be engaged. It will stay a couple of weeks, get bored. It will leave, go into another tissue, and it will stay there as well. Now, let's imagine another Treg. It only recognizes a component of myelin. Yeah? Myelin is essentially only found in the brain at any decent level. That Treg might pop into the liver. It won't see anything through its TCR, stay a couple of days, and leave. Then it pops into the brain, sees the antigen through its TCR, stays a couple of weeks, couple of months, then it leaves, and it circulates around. So that particular Treg, it might go through multiple different tissues, but the only tissue it will stay in for a long time will be the brain. So which TCR a Treg's got dictates which antigens it sees, which will dictate sort of where it spends its time across multiple organs, across one organ. Now, of course, the antigens that a Treg will see will also change during inflammation. The healthy brain and the inflamed brain present different antigens, so it might be a different set of Tregs that respond to damage in the brain compared to the Tregs that respond to a healthy brain.
Yeah, great. Thank you, Adrian. I guess one more quick question here, and then we can close out today's webinar. You're now looking at different genes for homing and so forth, but you mentioned you can, with this model as well, you could probably harvest the mice at different time points and so forth. And when you would see maybe what you think is going to be that, I'm trying to remember, I don't think you necessarily showed this data, a Treg that tended to be gut homing, and you know what that TCR is, or that flow code, you can probably see some of those TCRs that are in kidneys, for example, or other tissues that they maybe haven't, as you alluded to, get to their final place that they want to get to. And have you been able to explore, like, is any genetic differences or any different aspects from those that made it to their target tissue or where you think it is versus those that were transiently, maybe we think transiently passing through a non-specific tissue?
Yeah, great question. I mean, this is an area we're only just starting to go into. I mentioned at the end of my talk that there's an entry molecule for getting into the gut, and you see it a little bit in the mesenteric lymph nodes, but it happens in waves. So if you look really early on, you only see it being important for the gut. And then a bit later, when the cells start leaving the gut, you see it become important for the mesenteric lymph node. So the timing is absolutely critical. Now, on those recirculation experiments, some of those liver Tregs are going to end up in the gut, and some of the gut Tregs are going to end up in the liver. And interestingly, the liver Tregs that end up in the gut start looking a lot like gut Tregs, and the gut Tregs that end up in the liver start looking a lot like liver Tregs. So there is actually a lot of plasticity in terms of what phenotype that you see. But again, these are early days for these experiments.
All right. Yeah. Thank you very much. And again, I really appreciate you coming in here today and discussing this great new approach to do this massive parallel in vivo screening, right? Like these things are now applicable to numerous type of cell populations, and looking forward to seeing what other people do, and in terms of application of these retroviruses and what people can explore. So with that, on behalf of the ISAC community, we would also like to thank everyone who joined our presentation today. So if you're interested in participating in other incredible learning opportunities that CytoU offers, please check out archived Cyto recordings and on-demand webinars. We also greatly encourage viewers to participate in our online surveys once the webinar is concluded. Your feedback is very valuable in advancing our educational program. And this, to remind you, this webinar is also recorded and will be available to everyone. So please keep an eye out for more webinars in the future. Until then, thank you, Adrian, and have a great day.